Today I opted to go a bit more in detail for the technique of the sequencing, and so it will be less heavy on the neurodevelopment and a bit more towards the technical aspect. But I hope at the end of the seminar that you will be able to understand which questions can be answered, and the variety of questions that can be answered when we use the sequencing techniques that I will be talking about today. So today I'm going to talk about long read sequencing and its applications, for example, in neurodevelopment, because that's the field that I'm working in. And just for reference, I'm a postdoctoral researcher in the lab of Julien Cauchet at the University of Lyon. I've done my PhD in Belgium on cancer biology. And then I decided to move to neurodevelopmental biology because I got very interested in the brain, right? So let's start really from a very broad perspective, and that is something that I guess you all have heard about, and that's the central dogma in molecular biology, where you would have a molecule of DNA which can be transcribed into an RNA. And this RNA is then the blueprint that will give rise to a protein. It will be translated, and the resulting factor will be a protein. So this central dogma in molecular biology, you're all aware of that. Of course, the central dogma was at some point broken because there's something like a reverse transcription where you can also get DNA from RNA, but let's not go into that from now. The idea is here, and I will try to maybe put. Do you see a pointer? I will try to find my pointer. So you also see the pointer? Yes. Okay, perfect. Yes. So I was talking about the central dogma. Here you have the figure of the central dogma. So as I said, you get RNA from a DNA molecule. This RNA is the blueprint for proteins. And so basically all the information that you would have to make proteins would be somehow hidden, let's say, in the DNA and the RNA. So both DNA molecules and RNA molecules are very interesting for biologists to study to understand biological processes, although it's generally considered that the proteins are the actual actors in biology, right? However, as you also know, there is also a step between DNA and RNA, and that is the step of splicing. As you know, genes are consisting of coding sequences and non-coding sequences. Those coding sequences we call exons. The non-coding sequences we call introns. And so when transcription happens, the RNA polymerase will copy a DNA strand, resulting in what we call a pre-mRNA. And this pre-mRNA looks like has the same sequence as the DNA strand with the coding and the non-coding sequences. However, to get from a pre-RNA to a messenger RNA, which will be ultimately translated, something has to happen, and that process is called splicing. So splicing essentially is the removal of introns from a pre-mRNA and the joining of exons in that same mRNA. However, splicing for a certain RNA molecule does not always happen in the same way, right? Splicing of a same RNA molecule can give rise to multiple different mRNA molecules. For example, that's what you see here. In the first molecule, you have exon 1, 2, 3, 4, 5. While in the second molecule, coming from the same RNA, you only have exon 1, 2, 4, and 5. And in another variation, you only have exon 1, 2, 3, and 5. And the process of including exons differentially in a mature RNA is called alternative splicing. So alternative splicing is in some way a manner to build in diversity into the proteome with the same genome. You start from the same gene, you have the same pre-mRNA, but you can get many different proteins from the same mRNA. And that's what you see here, right? You kind of a protein A, a protein B, a protein C coming from the same RNA. And this is something that I'm very interested in, this process that we call alternative splicing, right? This is just a technical summary. So for splicing, you need two things. You need catalytic components, so the proteins and the RNAs that will actually remove the introns and join the exons. On the other hand, you also need a whole machinery that will actually define what is an exon and what is an intron. And those we call generally splicing factors. The details are not very important, but I just wanted to tell you this. And so, as I said, I'm very, very interested in alternative splicing. And as I said, I'm in the field of neurodevelopment. And I'm very interested in alternative splicing because in the last 10 years, it has been increasingly clear that alternative splicing is, first and for all, very, very important in the developing brain. The brain is the organ, together with muscle, where there are the most alternative splice forms found, right? And what people have defined is that during the development of the brain, there is a very precise temporal and spatial regulation of RNA isoforms, which means that in a neuron that has just been born, you will have a very different alternative splicing compared to a neuron that is in the cortex and that is doing its function. That being said, you have very different alternative splicing in an interneuron compared to an excitatory neuron, or in an astrocyte compared to an interneuron, right? And so this alternative splicing seems to be very much regulated during neurodevelopment, and so there are a lot of different things that we. So basically, we know that the process is heavily controlled, but we lack details on why it is important and how it is important. And as with all those biological processes that are important, of course, if something goes wrong in alternative splicing in the developing brain, you get disorders. I saw that one of you puts associated neurodevelopment with disorders. Well, splicing factors are very much enriched in molecules that give rise to neurodevelopmental disorders when they are mutated, for example. So this is actually my actual area of expertise. So this is what I'm very, very interested in, right? So generally, as I told you, DNA molecules and RNA molecules are very interesting for us biologists to study. And the way to study them is to do sequencing, right? And sequencing is essentially determining the order of bases in a given RNA or a DNA molecule. And here I give you a brief overview of all the developments in sequencing that have been done since 1977. So, of course, you all heard about Sanger sequencing, which was developed in 1977. Then there was the next generation sequencing, which was developed in or became commercially available in the late 2000s. And the sequencing techniques that we will be talking about today is generally called third generation sequencing or long read sequencing, who were developed about 10 to 15 years ago. And to really make you realize the impact and the techniques behind those newer forms of sequencing, I think it's important that I give you an overview of the sequencing methods and how they developed. So before I do that, I will make a small sidestep into another thing that you all have heard of, and that is the polymerase chain reaction. And why do I want to talk about the polymerase chain reaction? Because the polymerase chain reaction is often the conceptual basis of those sequencing techniques. So if you would make sequencing super simple, basically you're talking about a polymerase chain reaction. And I will be very brief because, as I said, I guess you've heard about this. But just in case you forgot or you did not hear about it, the polymerase chain reaction is used to basically copy pieces, segments, fragments of DNA. So how does that work? You have a double-stranded DNA. You denature the DNA into two separate strands. And basically, based on complementarity, you're going to copy those strands. And to be able to copy those strands, you need three components. The first thing is you need a DNA polymerase. That's the enzyme that's going to include bases based on a template strand. The second component you need are the bases themselves, the deoxynucleotides. So you have adenosine, thymine, guanine, and cytosine, right? And important is that they are deoxynucleotides. So you see this hydroxyl group here. This hydroxyl group is very important for the DNA polymerase to be able to put the next base. If this hydroxyl group is not present, the reaction cannot continue. So we have our DNA polymerase, we have our deoxynucleotides. The third thing we need is a primer, and this primer is important for the DNA polymerase to start the reaction, right? And then basically, as I said, based on the principle of complementarity between the bases, the DNA polymerase will do its job and it will copy the DNA strand. And then you start the process over again, you denature, prime, and the DNA polymerase does the rest, right? So that's very briefly the polymerase chain reaction. And here with Sanger sequencing, the first sequencing and the original sequencing method that I'm going to discuss, you will immediately see why it's important to understand the polymerase chain reaction, because the Sanger sequencing is basically a variation of the polymerase chain reaction. As with the PCR, you start with a double-stranded DNA that you're going to denature. If you want, you can make multiple copies to make the reaction more efficient. So that's a classic PCR. However, to perform the sequencing itself, what you're going to do is you're going to not only add to the reaction deoxynucleotides, but you're also going to add deoxynucleotides. And as I told you before, this hydroxyl group here is very important for the DNA polymerase to add the next base. However, if you use a deoxynucleotide, this hydroxyl group is not present, so the reaction will be terminated. That's why Sanger sequencing is also used chain termination polymerase chain reaction, chain termination PCR. And so if you have, let's say, 4 different tubes, all with different DDNTP, so let's say purple, you have a DDNTP variation of adenosine, yellow will be thymidine, cytosine, and guanidine. And that means that you can finish your reaction depending on the base here. So if you use this mixture with deoxynucleotides and deoxynucleotides, you will get a random incorporation of this deoxynucleotides and a random termination of your polymerase chain reaction. And so you will have a mix of very different fragments, which you can separate by electrophoresis. And basically then, based on the size that you have, you read out your sequence. In principle, it's very simple, but you can also understand that you can do this for, let's say, one strand of double-stranded DNA, one small segment, right? And then you have to do your electrophoresis, and it takes time. So you can imagine that if you have to do this for a whole complex genome, that this will take ages, right? It's not very efficient. So although this is the original sequencing that has been done for a very, very long time, to use it as a high throughput technique, it's a lot of work. So people worked on a way to bypass the limitations, the high throughput limitations of Sanger sequencing. And that brings us to the next generation sequencing. And basically next generation sequencing developed in the late 2000s. And there the important thing is that, or the thing is, there's lots of variations of next generation sequencing. You have different brands, different companies that developed their own way to do high throughput sequencing. Today, I will just give you one example, Illumina, because Illumina generally is the sequencing technique that would be used the most. So as opposed to Sanger sequencing, where you would start from a certain segment of DNA that you're particularly interested in, for Illumina sequencing, you can start from a bunch, a mix of DNA molecules. And basically what you do is you're going to put those DNA molecules on a certain surface, and the surface we call, in technical terms, that's the flow cell, right? And how are these DNA molecules going to attach to the flow cells? Well, you add what we call an adapter. So on each side of such a DNA molecule, you add a universal adapter. And so if you put your mix of DNA with adapter on the flow cell, that means that in a random way, those DNA molecules will bind to your flow cell. So you have like patches of DNA that are bound randomly to your flow cell. And then to have a reliable readout, what you're going to do is you're going to do a PCR, basically, on your flow cell. It's called a bridge amplification, but it's the same principle as a PCR. You use a polymerase to amplify the DNA strands, and this creates some kinds of clusters of all the same strand. That's what you see here. You see here there are 4 clusters of DNA strands, all with the same sequence, that are shown here. And after you've done your amplification, that's where the real sequencing step comes in. And that real sequencing step basically works in a very similar way as Sanger sequencing. You have some kind of chain termination, right? The difference here is that you use fluorescently labeled bases. So for example, here the guanidine will be yellow, the thymidine will be green, the cytosine will be blue. And the other big difference with Sanger sequencing is that the chain termination is reversible. So basically what you do is you have a first step of the polymerase incorporating a base, right? So that's what happened here. You have a cluster. And here the first base that's incorporated is the guanidine. And how do we know that it's a guanidine? Well, it's fluorescently labeled. So there's a laser, it excites the guanidine, and then you have your readout. For the other cluster, it's a thymidine, a cytosine, adenosine. What happens then? Then there is a chemical step in which the base that you see here, the fluorescence is removed, the fluorescent label is removed, and the base is then modified to be able to allow the DNA polymerase to actually incorporate another base. So it's basically a chain termination reaction, but one that is reversible and where there can be another step. And so one by one, after every chemical step, you have the new incorporation of another base. And that's basically how you determine your sequence. So that's basically what you see here. You have, for the left cluster, a guanidine, cytosine, thymidine, guanidine, adenosine, right? And the high throughput concept comes from having different clusters on the same flow cell, and all those different clusters can be read out at the same time. So that's how Illumina sequencing is working. As I said, it's one of the more common sequencing or short read sequencing methods, at least. Okay. However, I have not talked to you about what's actually happening before. So both for Sanger sequencing and for this next generation sequencing, there is an optimal length for those sequencing methods, and that has to do with the technical limitations of the machine that we have. And basically, this length of DNA is approximately 500 base pairs. That would be a good length to sequence. So what happens here, before you start with your DNA, basically in a chemical, enzymatic, or physical way, you make your DNA into small parts of 300, 400, 500 base pairs, so that you can put them in the machine and so that the reaction is efficient, right? So generally, what we say is that these techniques are short-read sequencing, because you cut up your DNA molecules or your RNA molecules into shorter pieces. So what happens is that your readout will be small sequences of 300, 400, 500 base pairs. So to make sense of those sequences, we need to map them onto the genome. And that's what you see here, right? You have all these different sequences. And all these different sequences, sometimes they overlap, sometimes they don't overlap. But basically, they're like small pieces of a puzzle that you have to fit into the puzzle itself, right? And if you take all those pieces together, you can make the genome, right? However, every piece by itself is only 500 base pairs. And that is directly related, so this short sequences, that's directly related to the pitfalls that you have in short read sequencing. So for example, if you have no reads in a genomic area, you are not able to map anything on that genomic area, right? So if you have a gap in the genome coverage, that's knowledge that's missing, and you cannot fill it in any way, right? Another pitfall is when you want to do pseudogene detection. A gene and a pseudogene, or a gene and its pseudogene, are very often they have a very similar sequence. So let's say if you have 500 base pairs, those 500 base pairs can fit in the gene, but they can equally fit in the pseudogene. You don't know. It's information that you do not have. Third limitation is related to the same principle. If there is a repeat region in the genome, you can map your sequence of 500 base pairs to maybe 1 or 2 repeats, but you have no clue whether there are 5 repeats, 20 repeats, or 200 repeats, because the sequence in essence is the same. And since you only have 500 base pairs, there is no way to know where it goes exactly on the genome. And the last pitfall is that you've all heard about epigenetics, right? It's the modifications of DNA without altering its sequence. Very famous, you have DNA methylation, for example. Since we are synthesizing in those sequencing methods, we are synthesizing DNA by ourselves in the machine, all the modifications that were present on the original DNA are not detected, right? So those are a number of very important pitfalls in short read sequencing, right? Okay, just are there any questions about this part already? You're still there because I cannot see you.
We are. 47 people, yes.
Okay, excellent. Yeah, since I'm having the full screen mode, I don't know whether I'm talking to myself. Yes.
Yes. This
no,
is
no, we're listening here. No.
Okay, perfect. So from time to time, I will ask.
Okay.
Good. So short read sequencing. I told you the pitfalls that there are with short read sequencing, and so to overcome those pitfalls, people have been thinking about long read sequencing. Right? They have developed a technique, third generation sequencing or long read sequencing, to overcome the problems that are with short read sequencing. And as you might guess from the name, long read sequencing will use longer fragments of DNA or RNA to sequence, basically to gain more information. Right. So I've talked to you about, mainly I've been talking to you about DNA at this point, but I'm very interested in splicing, which is on the RNA level, right? So just as a technical note, if we want to sequence RNA, basically what we are sequencing very often is not the RNA directly, but we do a reverse transcription reaction where we will use an RNA molecule to produce DNA, copy DNA. And it's this copy DNA that we are going to sequence afterwards. So generally, most of those sequencing methods are developed for DNA. And if we want to sequence RNA, we have to convert the RNA first into copy DNA, and it's this copy DNA that we're going to sequence, right? So, long-read sequencing. The principle of long-read sequencing is exceedingly simple. Basically, if you have one DNA or RNA molecule, you read that DNA molecule from the beginning to the end. No need to cut it up into different pieces. You start at the beginning and you finish at the end, and you read your molecule in one go, right? It's basically like a sentence. Where short read sequencing, you would read word by word. For long read sequencing, you would read the sentence in one go. Okay, so one molecule, one read, right? And just to give you an idea of what is possible, the current record for long read sequencing is 4 megabases. That means 4 million bases. If you compare that to the 500 bases that you get for a typical short read sequencing, the difference is enormous, right? We agree. And so in theory, the limitation of long read sequencing is your ability to get the longest molecule possible. If you could isolate a whole chromosome in its entirety in a way that is sequenceable, you would, with long read sequencing, be able to sequence it beginning to end. That's the theory. However, there are always practical constraints. So for now, the record is 4 million bases. And there are people that are trying to, I mean, basically there are labs and researchers who make it a sport to beat this record. Okay, good. So if we dive a bit into the technicalities of long read sequencing, basically for long read sequencing, there are two big players that have a commercially available long read sequencing technique. The first one is PacBio sequencing, also known as single molecule real-time sequencing. The other one is nanopore sequencing. Both are able to do long read sequencing with a reasonably similar outcome. However, the way in which they work is very, very different. And so I will guide you through the principles for each one, one at a time. Okay, so let's start with PacBio. As I told you, PacBio uses single molecule real-time sequencing. What does that mean? It means that in a PacBio flow cell, what you would have is you have all these small wells here. And at the bottom of such a well, you have a DNA polymerase that is fixed, right? It cannot move. It's there. It's at the bottom. It will not move. And the principle of this long read sequencing is basically if you get your DNA fragment that you want, what you do is you ligate some adapters to that on both ends. And these adapters are loops. So that means that you basically generate a very big circular molecule. And the circular molecule goes into a well. You have one of those circular DNA molecules per well. And basically the DNA polymerase will start copying the circular DNA molecule. And since it is circular, the loop keeps on going, right? So this DNA polymerase will be able to synthesize the strands over and over and over again, right? So how does the readout work? Well, the readout works very similar to Illumina or to fluorescently based Sanger sequencing. For every time that a nucleotide is incorporated, there is a laser that excites the nucleotide, which is fluorescently labeled, and the emission is detected. And that's the way you have the fluorescence readout. You can see that here. There is a cytosine that's incorporated. It is excited by the laser. The emitted light is red. The fluorescent label is discarded, and the cycle can start again. So you have a C pulse, an A pulse, and so on and so on. And so based on the fluorescence, you get the sequence of your molecule, right? The strength of PacBio smart sequencing is that you sequence this molecule over and over and over. And in that way, what happens is that the first time the DNA polymerase, I mean, it's going to make a number of errors, right? Biology is not perfect, so the DNA polymerase will include sometimes a base that's not the correct one, right? However, maybe in the second time, so it's a loop, so at some point it will come back to the same place. Maybe in the second time it will include the right one, and in the third time as well, because it will make another mistake at another place, but rather not at the same place. And so because you're sequencing the same molecule over and over again, you reduce the margin of error that you would have compared to if you would do the amplification only one time. But so important to understand is that the smart sequencing works with DNA polymerase. It includes fluorescently labeled bases, and the readout of your sequence is basically the fluorescence of those bases. And the thing is that you keep on going because you have made loops in your DNA molecule. Okay, so that's the first type of long-read sequencing. The second type of long-read sequencing works with a very, very different concept, and that's nanopore sequencing, basically. So with nanopore sequencing, the idea is that you have all kinds of small pores, and the pores are in the nanometer region. That's why it's called nanopore. And these small pores, they span a membrane. It's not so different from what you would have with an ion channel. And so basically here in the surrounding of this pores, there are electrolytes. And so basically you have some kind of ion current that goes through the pore. So if the pore is empty, you would have a basic ionic current that's going through the pore, right? At the top of this pore, you have a motor protein. And this motor protein is important because it will push, in this case, a DNA molecule through the pore. And you can imagine that normally if you have ions going through the pore, if there is something else in the pore, these ions will not go through as easily based on the size of what you put inside the pore, right? And so you will have a disturbance of the ionic current here at the mouth of the pore. And based on the exact disturbance of the ionic current, you can have a readout of your base. So for example, if there's an adenosine at the mouth of the pore, you will have a small disturbance of ionic current. However, if you have a cytosine at the mouth of the base, at the mouth of the pore, you will have a bigger ionic current. And so basically your readout is an electrical signal that fluctuates depending on what bases are at the mouth of the pore. And then you have an algorithm that is able to interpret those electrical signals and convert them to a sequence. So in essence, you have a motor protein that pushes your DNA strand through the pore. This disturbs ionic current at the mouth of the pore, and these disturbances are measured and converted into a sequence because the different bases in your DNA molecule disturb the current in a different way. Okay, that is a very, very different. That's a very different principle than the PacBio technique. Okay, so those two are at this moment the long read sequencing techniques that are the most developed ones. Okay, good. So does long read sequencing not have any pitfalls? Well, of course, long read sequencing has pitfalls, and that is the accuracy. Right, it has, especially with the first iterations of long read sequencing, there was a problem with accuracy. For example, with Illumina sequencing, so the next generation sequencing, what you would have is an error of 1 error per 1,000 bases that are read, which maybe does not seem like a lot, but if you read, I don't know, 20 million bases, the amount of errors start stacking up. Of course, this is a very raw readout, so that would be really the biological reality of what happens. And then with bioinformatics and with making a consensus sequence, you would be able to get this error rate down, right? However, if you use long read sequencing, the error margin is 10 times higher. Instead of 1 per 1,000, you would have 1 per 100 bases. And again, with bioinformatics, you can bring this error rate down. However, it is still inferior to what next generation sequencing can do. So if you use long read sequencing, this is something that you that is important to take care of, right? Okay, good. So, are there any questions about the long read sequencing and the principles behind it? No. Okay, good. One of the things that I haven't talked about is cost, and that is actually because next generation sequencing and long read sequencing have at this moment a very similar cost. It's not that one is much more expensive or whatever. So in that sense, it would be as efficient to do or cost effective to do long read sequencing compared to short read sequencing. Okay. Then we can move to applications, right? And since I'm a scientist that's interested in fundamental processes, I will start talking about fundamental research first. I know that the idea of the course is to give you an overview also how these techniques can help in providing a more to help with drug discovery and maybe some more translational research. But I think we can all agree that we first have to understand those processes, the fundamental processes that happen in biology, before we can find therapies. And this is especially true for the brain. I think the word clouds showed that you all think, and I agree, that the brain is an exceedingly complex organ. And so it's important to recognize that a lot of work has to be done still for fundamental processes in the brain before we can arrive to understand it better. But that being said, an important application of long read sequencing is fundamental research, right? How so? I will give you an example of my own interest field. So I told you I'm very interested in splicing, and specifically in alternative splicing. And splicing is something that you can look at with short read sequencing, right? It's typically done with sashimi plots, as they called, and you can appreciate that that it's called sashimi plots by the way they look when you represent them. So, for example, here, let's only focus here on this cassette exon part. So if I want to understand whether an exon is alternatively spliced, I can do that with short read sequencing. So what I would have, or what I would need, is reads that fall in between the boundary between the first exon here and the second exon. if I have a read that overlaps those two boundaries, I can confidently say, okay, those two exons are in an mRNA molecule. They are next to each other. Okay, and that's what you see here in the sashimi plots. From this experiment, I have 40 reads that span the exon-exon junction between the first exon and the second exon. On the other hand, between the second exon and the third exon, I have 25 reads that span the exon-exon junction. So what information can I get from this? I know that exon 1 is next to exon 2 in the mRNA and that exon 2 is next to exon 3. I also get some other information because there is also the case where I have reads that span from the exon-exon junction of exon 1 to the exon-exon junction of exon 3. And that's what we see here. So I can infer that there are at least two isoforms of RNA present. One RNA isoform with exon 1, 2, and 3, and one RNA isoform that has exon 1 and 3, but that lacks exon 2. However, I do not have any information whether exon 1 is present in the same molecule as exon 14, because with my limited amount of bases, my short fragment of 500 base pairs, I cannot find a junction between exon 1 and exon 14, right? So basically with short read sequencing, you can have information about exons that are next to each other, but you don't know what is happening with exons that are further away. I have no clue whether exon 1 is in the same molecule as exon 14, or whether exon 2 is in the same molecule as exon 6, right? I can make a statistical inference of that by looking at the different exon junctions and the different data, but in principle, I do not know. And that is a very big shortcoming for short read sequencing when you want to look at splicing, right? So here, what we would do with short read sequencing is we would kind of have some kind of guess of what is happening with the mRNA isoforms. However, with long read sequencing, I will sequence my RNA from the beginning to the end. So I know that exon 1 is next to exon 2, it's next to exon 3, it's next to exon 4, it's next to exon 5, it's next to exon 6, right? And in this molecule, for example, I can see that it starts with exon 3. It does not have exon 4, but it does have exon 5 and 6. And this kind of information I would completely lack with short read sequencing. So that's why I'm very interested in long read sequencing, because it gives me the full overview, in theory, it gives me the full overview of all the RNA isoforms that are being expressed at a certain time in a certain type of cell, right? So this is it gives me much more information, right? And this is a very nice paper that I will not go into the details, but that came out last year, where they did long read sequencing. And they looked at splicing patterns in single cells. And so, for example, the information that you can get from that, if you do a proper bioinformatics approach, is you can look at all the isoforms of RNA that are expressed. And here, in this case, they looked at different types of neurons. They looked at excitatory neurons versus interneurons. They looked at neuronal subtypes, and they looked, so for example, neurons in the first layer of the cortex versus neurons in the sixth layer of the cortex, and they also looked at age. And so basically, you can get a lot of information from here. So for example, here they show that excitatory neuronal subtypes are very much enriched in specific isoforms. That's what you see here, but that these isoforms do not change so much with age or with cell type. Okay, so it's a bit technical. The paper is really great, but this is the kind of information that you can get with long read sequencing. Okay. A second application that is more in the realm of fundamental research is if we want to look at epitranscriptomics. I think most of you have heard of epigenetics, but I think maybe not so many of you have heard about epitranscriptomics. However, it is very analog to epigenetics in the case that where epigenetics are modifications of the DNA without altering its sequence, epitranscriptomics is the study of RNA modifications. And here, as you can see, there is an RNA molecule, and there are a number of RNA modifications that are present in this figure, but the reality is that there are more than 120 RNA modifications that are known, and the vast majority we do not know what they do. But what we do know is that these RNA modifications, like DNA modifications, alter the expression of genes. RNA modifications alter the fate of RNA. When some modifications are present, an RNA will be more translated. When some modifications are present, an RNA will be more degraded. And that's exactly what we see here, for example. Like, again, very analogous to epigenetics, you have readers and writers and erasers of RNA modification. So if we start with an adenosine here, this is an unmodified RNA or an unmodified RNA base, right? And you have these enzymes here, methyl 3, methyl 14, methyl 16, which are able to put a methyl group on the nitrogen, on the amine group here. So they add this extra methyl group, right? Those are called RNA modification writers because they write the RNA modification. On the other hand, you have RNA modification erasers, ALK-BH5 or FTO, which are able to remove this methyl group again, right? So basically you have enzymes that put a methyl group or that remove a methyl group. However, what is the effect? Well, you have readers of those RNA modifications. In this case, for N6-methyladenosine, it will be YTH proteins. And you can see that if you have a methyl group on your adenosine, this means that YTHDF1 can bind and translation will be repressed. On the other hand, YTHDF2 can bind and your RNA gets degraded. So we know that these RNA modifications are important and that they might alter the fate of an RNA, right? So how are RNA modifications usually looked at? Well, basically one by one. So for example, if we look at methyladenosine sequencing, what happens is you purify your RNA. As you can see, there are some methyladenosine bases present, right? You fragment your RNA because that's what you do in short-read sequencing. And then basically what you do is you have your input control where you will sequence all the RNA that you have. And then from the same pool, you will immunoprecipitate with an antibody the methyladenosine residues, and you will sequence just this pool of methyladenosine sequences, right? You put them together and you have an idea about which bases are modified. However, you only looked at the methyladenosine here. The same goes for pseudouridine. It's another RNA modification. It works slightly different, but the concept is the same. You have the CMCT. The CMCT will react chemically. It will do a covalent binding with pseudouridine. And if the CMCT is present, it will not allow the reverse transcriptase. If you do a reverse transcription, it will block the progression of the reverse transcriptase, which means that every time you have a pseudouridine, the reverse transcriptase stops. And if you sequence this, every time it stopped, you have your places where there was a pseudouridine before. But again, you only look at your pseudouridines. However, as you can imagine, an mRNA does not only have pseudouridines, it does not only have methyladenosines, but it has a lot of different modifications at a lot of different places. And you can imagine it like this. If you do this sequencing one by one, it would be as if you are reading a book, and in terms of punctuation, you would only look at the exclamation marks, right? Or here you would only look at the question marks. And maybe for another type of sequencing, you would only look at the full stops. If you only do that, probably the book is not going to make a lot of sense, right? You need all the punctuations to really understand what is going on, what the text is saying. And it works the same for an mRNA. If you only look at one modification at the same time, it will likely not make a lot of sense. So there has to be a way to look at multiple modifications at the same time. And that is where long read sequencing comes in. And I have to say it's specifically long read sequencing with nanopores, because this does not work for PacBio. And that is because of the difference in concept by which they work. In the sense that if you use nanopore, as I said, it's a disturbance of the current at the mouth of the pore. And I told you that different bases at different times, different bases have different disturbances of the current, right? However, you can imagine that if you have a modified base, that this modified base will have a slightly different disturbance of the current compared to an unmodified base. And that is exactly the principle that is used, or that you can use to look at RNA modifications using nanopore, right? Here, so what you would see here is an electrical current pattern for an unmodified base in red, right? And you can see where the bases are modified. For example, here in the blue line, there is a difference between the blue and the red, which means that this base is going to be modified. So where PacBio, since it's working with the DNA polymerase, you can only use DNA or copy DNA there. However, nanopore works with the disturbance of the current. So what you can do is you can directly take your RNA and you can put it on the pore without making DNA first. And since you are using your original RNA, it means that you keep your original modifications, which you can read out. So the limitation in this case for modified bases is technical, in the sense that it depends on your signal to noise ratio for the readout of the current. That's the first thing. And the second thing, it depends on how well your algorithm is able to distinguish the differences in current. Right. For now, nanopore is working on detecting 2 or 3 modifications at a time. But in theory, it's possible to look at all modifications at the same time. Okay, so that's very, very exciting. Okay, is there any questions about what I just explained? You're still there?
Yes, we're there, we're listening, but it's very complicated.
Oh, it's complicated. Okay, so do you have questions?
No, I think students maybe have some questions. I don't know, maybe they are shy.
Okay, I mean, we can also do the questions at the end, but...
Hello?
Hello?
Hello, my name is Angela.
Okay.
Nice to meet you. Thank you very much. So, is it important the ion or the ions you use in the nanopore for the current? Do you understand my questions?
No, I do not understand your question. You mean
Is
the
important
type of
the
ion
ion?
that you use?
Yes.
Yes.
It
So
modifies
that is
the result?
yes, because you can imagine that if you use different kinds of ions, the current through the pore will not be the same. A calcium ion will not behave the same as a sodium ion, will not behave the same as a potassium ion. Honestly, I am not sure if they use one type of ion or a mix of ions or whatever, but this is standardized. So you cannot use a solution that you made yourself and use it with that. No, it's provided by the company. And if you would use another solution, the pore will behave differently, right?
Thank you.
Yeah. Any other questions? No, not for now. Okay, good. So that being said, that's for the fundamental research part. Okay. However, if we go a bit towards the clinic, long-read sequencing can also have an important impact in terms of diagnostics, right? So I told you before that one of the pitfalls of short-read sequencing are repeat regions, for example. And we call these sequences that, or the genomic regions that are not easily mapped by short-read sequencing, we call them dead zones. So let's assume we have a genomic region with 6 repeats, 1, 2, 3, 4, 5, 6 repeats, right? And we do short-read sequencing. What we can see at most from the bioinformatics will be 2 repeats, because we have some sequences that are inside the repeat. Okay, that's 1 repeat. And we will have some sequences that are on the verge of 2 repeats, on the junction of 2 repeats, which means that we know that there are at least 2 repeats together. We know that the repeats exist, but we have no clue whether we have 2, or in this case 6, or based on short-read sequencing, we might have 20 or 200. We simply don't know. However, with long-read sequencing, we can sequence those dead zones, right? Because we start here and we will read the molecule from beginning to end. And in that way, we know there are 6 repeat regions. So there are some disorders that are related to genomic repeats. Now there are relatively labor-intensive and cumbersome methods to know how many repeats a patient has, but with long-read sequencing, this would be much more easy. Okay, and so here I brought you a small overview of what long-read sequencing can mean in terms of diagnostics. So as I said, there is structural variation, so the number of repeats. You can make the distinction between a gene and a pseudogene. Let's say, even if they have the same sequence, since you have the whole molecule, you can easily distinguish them. Repeat expansions, rare variant identification. But what I want to talk to you about is imprinting disorders. And imprinting disorders, or imprinting as a biological process, I'm not sure that you've heard of it, but I might think yes. So basically, you know, for all the autosomal chromosomes, you have 2 alleles. You have 2 copies of the same gene. And in most of the cases, both of those alleles are used for transcription and expression of your mRNA, right? However, there are some exceptions. And those exceptions are imprinted genes. That means that the genes on one of the chromosomes, either the paternal one or the maternal one, will be inactivated. So we talk about maternal imprinting when the maternal chromosome is inactivated, which means that those genes are only expressed from the paternal chromosome. You can have the other way around, paternal imprinting, where the paternal chromosome is inactivated and genes are only expressed in a maternal way. So this whole process of inactivating genes on one set of autosomal chromosomes is called imprinting. Okay, and there are a number of imprinting disorders, and I give you here the example of Prader-Willi syndrome. Prader-Willi syndrome is an imprinting disorder. There are some very obvious clinical telltale symptoms. So, for example, when those babies are born, they have muscle dystonia, so they cannot keep themselves upright. You can see that something is wrong with the muscles, and throughout, let's say, throughout their development, they will develop craniofacial abnormalities. There's often a bit of intellectual disability. You can have self-harming behavior, autistic spectrum disorder-like behavior. There is hypergonadotropy. So it's a syndrome with a lot of symptoms, and you can imagine that it's not easy for those patients. And in terms of if we look at what causes the Prader-Willi syndrome at the genetic level, there are 3 possible causes. I told you it's an imprinting disorder. So one of the things is that normally those genes in Prader-Willi syndrome are on chromosome 15, and normally they are only expressed from the paternal side. So normally, as you know, you get 1 chromosome from the mother, 1 chromosome from the father. Normally, they're only expressed on the paternal side, and they are silenced on the mother's side. However, what sometimes happens is there is a mistake, and they're also silenced on the paternal side. So that means that those genes are neither expressed from the maternal side, neither expressed from the paternal side, and that causes Prader-Willi syndrome. Very similarly, if you do not have a chromosome, if you did not, for one reason or another, inherit a chromosome from mother's side, from the paternal side, you have 2 chromosomes from mother's side, and both chromosomes are silent. You will not express those genes. A third reason can be that you just lost the region of the paternal chromosome where those genes are expressed. So the mother's side is silenced, and the deletion is at the father's side. So basically, if in any way you lose or you do not express those genes on the paternal chromosome, you develop Prader-Willi syndrome. Okay, and here I bring you a scheme of the diagnostic strategy for Prader-Willi syndrome. So as I said, there are some telltale symptoms when a baby is born, where a doctor might say, I think this child might have Prader-Willi syndrome. And so the first thing they do is a methylation analysis, right? They look at this region of the chromosome, chromosome 15, whether it is methylated, yes or no. And so basically what you get is you start with the methylation analysis, then you have a FISH, then microsatellite analysis, imprinting center analysis, and so on and so on. So basically, if you are unlucky, you have to wait for a very long time, and you have to do 4 diagnostic tests before you know that your child, or before the doctor knows that the Prader-Willi syndrome is present, and what is the cause of the Prader-Willi syndrome. However, with long-read sequencing, you could do the diagnosis of Prader-Willi syndrome in one go. Why? Because if you sequence it, you can see if the region is deleted, yes or no. You can also make the distinction between the 2 chromosomes. So if you have 2 chromosomes from mother's side, you will also see that with the sequencing. And thirdly, you can also look at the methylation of the DNA sequence, because you can make the distinction between modified and unmodified bases. So if you would use long-read sequencing, all this process with those 4 tests basically would not be necessary anymore. And so I give you now the example of Prader-Willi syndrome, but there are a lot of diseases where genetic testing is still done with this kind of diagrams, which could be relieved, for example, by long-read sequencing. And that is, for example, what this Japanese group has done. They have developed a way to do long-read sequencing to diagnose Prader-Willi syndrome patients. And so more and more, in the lab, we work with a number of clinicians who are interested in genetic diseases, and they say that more and more long-read sequencing is going to be implemented in the diagnosis of patients. So this is a very interesting development in the clinic, and where the emergence of a new technique really can make a difference in terms of cost effectiveness. But also, you can imagine that if you have a child that is sick, you want to know as fast as possible what is going on. And so that this one test of long-read sequencing will be much faster than doing 4 different diagnostic tests, right? Good. So then, if there are no questions, we can move to the last part, and that would be the protocol. So I will outline very quickly the conceptual steps of protocol. So what I do, for example, is I want to look at splicing, and so I will use copy DNA with a reverse transcriptase with Oxford Nanopore technology, so with Nanopore. And so I will specifically go with you through protocol where we can do this. So first we have the conceptual steps of the sequencing protocol, starting with acquiring the biological material, isolating the RNA. Then there's the quality control. We do a library preparation, the sequencing, and in the end, the bioinformatic analysis. Okay, so I can be very short about acquiring biological material. You have to do it in a way that the quality of the tissue or the cells or whatever you're harvesting is preserved, because the quality of the starting material will ultimately determine the quality of the output. If you have a sample where all your RNA is already fragmented because you did not keep it on ice, it means that at the end you can do long-read sequencing, but if you have fragmented RNA, that doesn't change, or you will not have long reads, right? So be very careful with the quality of the material that you start with. Secondly, isolating RNA. There's no specific step for long-read sequencing. So it's the same type of RNA isolation as you would do for any other RNA sequencing method. So what we use in the lab routinely is columns. You have some some silica columns, which in the right conditions, they bind the RNA. You can do a number of washing steps where the RNA is still bound on the column. That's what you can see here. And in the end, you use an elution buffer to get rid of the charges of the silica column, and you elute your RNA. Depending on whether you want to keep DNA or RNA, you add a DNA step or an RNA step. If you want to keep the RNA, you get rid of the DNA using a DNase, and otherwise you use an RNase to get rid of the RNA. Okay, so isolating RNA is a very classic way. And then there's the quality control. I'm quite sure that all of you have heard about 260-80 ratio and 260-30 ratio. So if we put our solution of RNA through a spectrophotometer, what we want for RNA is a 260-80 ratio of 2. For DNA, we would use a 260-80 ratio of 1.8. And then the 260-30 ratio, that's for proteins and other impurities. We also want that to be around 2.1, 2.2. So that's the first way to look at the quality. Second way to look at the quality is to see whether your RNA is still intact. And that you can do by looking at ribosomal RNA. I'm not going to go into the details, but basically there are two ribosomal RNAs, 28S and 18S, and you can take the ratio. And if the ratio is intact, that gives the number 10. And you can see here that if there is degradation of the sample, you can get all kinds of other bands of degraded RNA. The 28S will not be present anymore. And so basically this really shows if your RNA is complete, yes or no. That's what you can see here as well. Here the RNA would be complete. Here it's kind of completely degraded. Okay, so that's for the quality control. And then there is a step of library preparation. That's maybe the step that is conceptually least that is conceptually least common to you. Library preparation is a step that is generally used in sequencing methods. And it is the process whereby the input, either RNA or DNA, is modified to suit the sequencing method. You remember from the Illumina sequencing, basically what you have to do is you have to put adapters on each side of the DNA to add it, or for it to be able to bind to the flow cell, right? And so all these steps that you add to make your RNA or DNA suitable for the sequencing method is called library preparation. In our case, this is a reverse transcription because we start from RNA and we are going to sequence the copy DNA, right? So in reverse transcription, we take an RNA molecule and we make a DNA molecule out of it. We have adapter ligation and addition of motor proteins. You remember that for the nanopore, there is a motor protein that is present and that pushes the RNA or the DNA molecule through the pore. Well, not all RNAs or DNAs, they would be able to bind the motor protein by themselves. So what you do is you include some sequences that are able to recognize those motor proteins, and then you add the motor proteins by themselves, right? So basically, it's a bit technical, and it changes for every sequencing method. If you want to sequence RNA, it's going to be a bit different compared to when you sequence DNA. If you use a nanopore, it's going to be different when you use a PacBio compared to a next generation sequencing. So library preparation is really dependent on the sequencing method. Generally, for library preparation, for long read sequencing, it's much more simple than what you would do for next generation sequencing. So that's one of the advantages. You induce much less error during the library preparation compared to next generation sequencing. It's not error-free, but you have much less errors, okay? Then next step is the sequencing. You put everything in the machine and you let it do its work. And then lastly, you have the bioinformatic analysis. I'm not going to go into details here because that's a whole topic by itself. But know very well that you have to know the limits of your bioinformatic analysis. And every sequencing technique has a specific output, and you will tailor your bioinformatic analysis towards that output. Okay, good. So that would be it from my side for now. So that's the theoretical part. And so from what I understood now is that we have to make a protocol together. That's right?
Yes, the students that will contact us can work in the group on the preparation of the protocol. So actually, once the protocol is ready, we will ask you, Martin, for the revision, and then revised protocol lands on the project website. That's the idea.
Okay.
Yeah.
Very good. So what I propose as a protocol, I mean, it's quite specific, would be to look at copy DNA through nanopore sequencing, specifically from the somatosensory cortex in the brain.